This webinar has been set up jointly by the European AIDS Treatment Group, PrEPster, AVAC, and PrEP in Europe Initiative, to look at the very, very interesting developments in the pipeline for innovative prevention methods. I've got a few technical things to tell people myself. Firstly, this is being translated into Russian, and there is a Russian translation channel. On your Zoom screen, if you're looking at this on a computer, you will see a channel, a sign at the bottom right-hand side of the screen, which says interpretation. If you're listening on headphones, click on the channel that you wish to hear, and you should hear simultaneous translation. And I've just checked, in the language of your choice, okay? So we thank very much the translators for that. I think Sarah has already said, this is a recording meeting, and if you don't consent to an image, then make sure your video is off. Speak slowly and carefully, and this applies to the presenters, but it also may apply later to people asking questions. Because of this, there are many people whose English is not their first language in this presentation, so make sure people can hear properly. I will generally let the speakers introduce themselves, but we have basically 3 presentations from Cindra, from AVAC, from Penny Moore of the University of Witwatersrand, and from Ferran from BCN Checkpoint. And we're looking very much forward to what they're saying. There will be feedback forms sent out by email after this session, and it will be published in a number of different places, including the EATG's website, AVAC's website, I imagine, and also on PrEP in Europe's dedicated YouTube channel, which you can access through our website and probably on PrEPster's, I imagine, as well. I want to do a quick plug for PrEP in Europe's next webinar, which will be on women in PrEP and is scheduled to happen on May the 15th. And I think that's all I want to say about the technology of this. Basically, just a couple of words. We are faced with a new generation of very interesting and very promising HIV treatment and prevention modalities, which are basically long-lasting, which involve injections and implants and things like that. These have the potential to plug the gap between daily treatment and what we eventually hope will become a vaccine, because they may involve being able to take an anti-HIV remedy or an anti-HIV prevention remedy at least only once a month, and maybe less often than that. And I think this is very exciting. However, we are faced with the same situation that we have every time a new HIV technology comes along, which is that of access and availability. And one of the key questions that will be addressed in this webinar is making absolutely sure that these very exciting possibilities become on offer to all the people in the world who need them. Having said that, I am now going to hand over to Cindra, who can introduce herself and will present the first set of slides, the first presentation. Thank you.
So just really briefly, I'm Cindra from AVAC. We are based in New York, and I am based in France. And our mission as an organization is to bring HIV prevention research and all of the experimental candidates through the research process, and to get successful products and to bring it to the rest of the globe to where it's needed the most. So today I am going to talk to you about the next generations beyond oral PrEP. So just to put it all in context, what's happening on a global level with HIV prevention, if you look down at the bottom of the screen, in 2010, we were at around 2 million new infections a year. And therefore, we projected, or UNAIDS projected out, that by 2020, we should, to get epidemic control, we should use all the prevention we have to get down to 500,000 infections no more than 500,000 infections in the year of 2020. However, we severely missed the target, and we are at around 1.7 million new infections each year, which is not a big difference from 2 million 10 years ago. So there's a lot of work to be done, both scientifically and socially, with implementation. Uh-oh. Okay, wait, okay. I'm gonna start with what we have. We've had about 8 years of daily oral PrEP implementation, and this is mostly in the form of TDF FTC, which is the brand name is Truvada. And it's also in some places in the US, FDA has approved TAF, which is a form of Truvada. So there's those two formulations. And there's also, there's not only daily PrEP, but intermittent PrEP or on-demand PrEP, which you take tablets before and after sex, are also part of the accepted PrEP regimen that is being implemented globally. And you'll see that there is about 3 million people on the globe who are eligible for PrEP use. But until just a few months ago, we only had 31% of these 3 million people on PrEP reaching this target. So there's clearly an issue of access, of availability, of desirability, acceptability. Why are people not taking PrEP? Why are they not getting PrEP? This is a table from a graph from 3 African countries showing that when people first took PrEP, the first month, people who stayed on it, it was under 20% of the people stayed on it in the first month. And we're looking at everyone from female sex workers to gay men, to young women, to serodiscordant couples. And then by month 6, people on PrEP were down to less than 3%. So there's clearly a problem with accessing oral PrEP and staying on oral PrEP. So to get around this problem is one of them. So the question is, why are people not staying on PrEP? Well, first of all, a daily pill is a big burden, as we know, especially for people who are not living with HIV. To prevent something, it's a big ask to ask people to stay on this pill. And so we've learned from the oral contraception field that the more options people have, the more unintended pregnancies there are. The more options people have with HIV prevention, the more unintended infections there are. So today we're going to focus on right here, these 2 areas. But first I want to draw your attention to what do we have right now to prevent HIV. We have treatment. So we know that undetectable equals untransmittable. We have condoms. We have voluntary medical male circumcision, which is actually only for Africa because it's only really works with men who don't have receptive anal intercourse. So it's really for heterosexual men, syringe exchange, and we have daily oral PrEP or event-driven PrEP. And then all the way in the beginning of the clinical pipeline, even preclinical, before it even goes into humans, there's a whole bunch of different ways of different compounds and delivery mechanisms that that science is working on right now as we speak. And as Gus mentioned, there's long-acting implants. For example, there's a phase 1 trial that shows that an implant loaded with the drug islatravir can stay in your arm for up to 1 year and hopefully prevent HIV. That means you don't have to think about HIV, or someone who's at risk does not have to think about HIV prevention for an entire year as it slowly releases from this implant. And then there's preventative vaccines. I'll just mention that a little bit. There's vaginal rings that get loaded with drugs that can prevent HIV, can prevent STIs, can prevent pregnancy. And now, keep in mind, these are all experimental. They haven't been proven to work yet. Broadly neutralizing antibodies that are passively infused, and Penny Moore is here to talk about those today. There's anal and vaginal inserts with drugs that can be inserted into either of the compartments, as they say. There's skin patches and even enemas. And these are all things that are being researched. But today we're going to focus here on the Depiving Ring and a long-acting injectable, because these were just proven to work, and they're moving along out of research into regulatory approval. And I'll also cover a little bit about what's in large-scale studies. So these have made it all the way through phase 1, phase 2. Now they're in phase 3, and what it means if they are proven to work, they could go to the market soon. And so again, we've got new long-acting injectables, preventative vaccines, daily or monthly oral PrEP, and we even have a dual protection prevention with PrEP and oral contraception. So I'm going to just focus on these. So these are the late stage efficacy trials, and these stars mean these are the times when we might actually see these products go to market. So there's the vaginal ring, and I'm going to talk about the details of the vaginal ring, but it was just approved by the European Medicines Agency last year, and it's actually going to be moving into some African countries at the end of this year. And then we've got long-acting injectables, and I'll go into the Kabbategravir long-acting injectable that is the new shiny toy that everybody's talking about. It's a 2-month intramuscular injection, and it can protect people for 2 months. And in fact, it's superior. It works even better than oral PrEP. And then further, and that's actually could come to market within a year, or not within a year, in a year. And then we've got lenacapavir, which is an injection every 6 months. So that means twice a year you get an injection, and that is just starting in the clinical in the phase 3 clinical trial, and it could be ready as soon as 2024. I'll talk a little bit about the dual prevention pill, which combines TDF FTC PrEP with oral contraception for women. We could see that as early as the end of 2022. We're looking at TAF. TAF is another oral. As I mentioned, it's an alternative to TDF FTC, and it's been proven to work in men who have sex with men and trans women, but it's never been studied in cisgender women, and this is about to happen now in women in sub-Saharan Africa. And then there's also islatravir, which is another type of ARV, and they're looking at this as a monthly pill. So you would take this monthly to prevent HIV. And then lastly, we have preventative vaccines, and this is the adenovirus 26. It's a flu-like virus, and they insert all different synthetic clades of HIV from around the globe. And this is in a late stage trial as well. And if it is proved to be effective, we could possibly see it at market as early as 2024, which is amazing. And not only is that, it's the only HIV vaccine that's ever been studied in Europe. It's being studied in Poland, Italy, and Spain. Okay, so let's get into the Depiving Ring. So the Depiving Ring is about that big, as you see in the picture. It's silicone, and it's initiated monthly by a woman. She learns how to initiate it herself. It's discreet. Nobody can really detect, the sexual partner cannot detect it's there. It doesn't interfere with sex. It's been proven to safe. And we call it non-systemic, meaning it's not flowing through the bloodstream. It's the drug stays in the vaginal compartment, and it doesn't go all around the body. It just stays where the HIV might be transmitted. And Depivering is the ARV drug that's loaded and slowly released over 4 weeks. And after 4 weeks, you get a new one, and you use it if you need it. The thing with the vaginal ring is that it's not as protective as oral PrEP. It has been shown to be anywhere from around 35 to 60% risk, reducing risk. Now, This goes a long way in some African countries where it's actually been, where the WHO and the European Medicines Agency has actually approved it or recommended for its use, only in Africa. The EMA did not approve it. It hasn't been, they have not sought approval for Europeans. So the question is, why not? Is this something that European women would use, given that it reduces risk by a lot less than oral PrEP? The reason why they think it's useful in Africa is because women can use it. It's acceptable to women, but also the rates are so high that it could actually, something that lowers risk by 40 to 60% could have a large effect on the population, even though it's not that great. So the question is would it be worth rolling out in Europe? And if so, advocates need to get on it and ask and demand that it be regulated and implemented in Europe. So the next is long-acting injectable Kabbategravir, and this is the sort of shiny new big deal that everyone's talking about because it works even better. It was shown to be statistically better than oral PrEP. It's a class of drugs that's the same as dolutegravir, which is a first-line treatment drug. And it's intramuscular injections every 2 months in the butt. Trans women can't get it in their butt if they have implants in their butts. And we know that trans women are at very high risk. So that's actually not a good thing. So there's 2 ongoing studies. We have the results, but we're looking now at open label. So we're following the study even more. The great thing about this is that 6 shots a year protect people. You don't have to think about taking a pill every day. And also this is already approved with another ARV, rilpivirine, for treatment. So this is just breaking down how well it worked. They compared the CAB long-acting injectable in this arm to the oral TDF-FTC. And it turned out that there was almost 3 times more infections in the oral PrEP arm than there was in the injection arm. And that's kind of it's kind of a no-brainer. They show that most of these infections, 37 of these infections in the TDF arm, was because people didn't have drug in their body. They never took their pills. And that's the whole point of getting an injection. You don't have to take your pills. There was, however, when they had long-acting injectable in their bodies, there were 4 breakthrough infections. So they're still not sure why how this happened. So they're still looking to see how it happened. But what they did see, they saw a lot of Kabbategravir resistance infection. And that's because the Kabbategravir was so good at fighting the HIV infections that the ones that were broken through were not detected with the standard HIV tests that look for antigens or antibodies. So one of the things that they're thinking is if this gets rolled out, perhaps they need more sensitive HIV testing to see if someone has HIV. Maybe they have HIV before they even start Kabbategravir. Anyway, the point is this is the first ever long-acting injectable to show really great results. Going forward, we need to understand why those 4 infections happened, but we're talking about 4 breakthrough infections in over 2,000 people. So still, it's a very, very small amount of people who got infected. Currently, we need to move in our countries to offer, they're offering the participants in the trial a choice between the Truvada or the Kabbategravir. They're going to be studying it in other populations, such as adolescents and pregnant women. It's being studied now in women in Africa. This study that I spoke to you about was studied in gay men and trans people. ViiV is the pharmaceutical company who manufactures it. They are applying all over the globe for all populations. to the FDA, the EMA, and in South Africa. And hopefully by next year, we will be starting to roll it out. And so what's the advocacy that we need around long-acting Kabbategravir? I mean, the science, they say, is the easy part. The hard part is getting these proven interventions to the people that need them most. And that's where we as activists really have to come in. So with this, perhaps we need better, more sensitive HIV testing when someone's on Kabbategravir to really know whether they're HIV positive before they start, or if they've seroconverted while they're on it. And this will avoid the drug resistance. And I said it's the same class as dolutegravir, which is a first-line treatment drug. So you don't want to knock out whole populations that can't take a first-line treatment drug. We don't know what the price is yet. It hasn't been priced. And it's only going to be cost-effective if it's no more expensive than oral PrEP. We need alternative injection sites for trans women who have buttocks implants. We need to, if we're going to roll it out, we need not just viral load testing or better testing, we also need an alternative to integrase inhibitors wherever it's being rolled out. A lot of resource-poor countries use dolutegravir as a first line. Mm-hmm. And we should quickly move to pilot programs for Kabbategravir in places so that we can learn as quickly as possible how to roll it out.
Cindra, we've only, well, you're overrunning, so if you can finish, I think you've got a couple of more slides left, that's all.
Sure, thank you, Gus. So the dual prevention pill, it's just a combination of what we already have, which is an oral PrEP pill, but it's combined in one tablet with oral contraception, and that would be really attractive to young women. I'm not going to go into the details of that. Let me skip ahead to, okay, here's in conclusion. We have more safe and effective prevention options beyond oral PrEP entering the market and in development, as I showed you. But currently, oral PrEP as TDF-FTC is the only PrEP that we have that's been approved with clear programmatic guidance, targets, product availability. Now is the time to integrate the Depiving Ring and the CAB long-acting injectable. And so what we can do as advocates, as frontline workers, is we can increase resource allocation, so increase the funding for HIV prevention to facilitate national scale-up of all of these PrEPs, whether it's new PrEP or the old standard PrEP. That doesn't mean take money away from treatment, but it means more money for HIV prevention. We need to develop plans on how PrEP can contribute toward epidemic control that incorporate these new HIV prevention modalities, such as long-acting injectables, or eventually implants and everything else. And we can engage in all different systems to move this ahead quicker and not wait like we waited, especially in Europe and especially in Eastern Europe. We don't want to get delayed like we got delayed with oral PrEP. We can engage with the regulatory approval processes in our countries, enabling policies and guidelines, service provider education, service delivery channels, and demand generation. And with that, I'll stop there. For those of you who do get my slides, this is a very busy chart, and it just goes into the details of everything I just said and more. And please, you could reach out to me at AVAC for any other questions. Thank you so much for that. Any clarifying questions?
Yep. Very briefly, any clarifying questions just for things like technical issues like acronyms and things, but we're a bit behind, so we will leave all other questions, and I see Nicholas has asked a particularly interesting one, until the panel discussion. And so I think we'll move straight on to Penny Moore, who is somebody that I remember from, oh God, the last HIV vaccine conference in Barcelona that happened before they turned them into HIV R4P. And she gave the most amazingly comprehensible explanation I've ever heard on the problems of doing an HIV vaccine and the nature of broadly neutralizing antibodies. So I'm delighted but she's speaking on exactly that subject now. So Penny, you can introduce yourself and take it away.
Thanks, Gus. I hope I can live up to that. And please shout at me if I start running over, because I know we're short of time. Hi, everyone. Yeah, my name's Penny Moore. I'm based at the University of the Witwatersrand, which is impossible to pronounce, in Johannesburg in South Africa. And I'm going to try and summarize at a very high level, because I think that's what this is probably about, where we are with vaccines and broadly neutralizing antibodies, and there's clearly an overlap there. So I'm going to speak today about really two major concepts. One is what we call active immunization, and the other is passive immunization or passive vaccination. Active immunization is old school vaccines, how we've always done it for polio and measles and mumps, where we give a little chunk of protein often to people that resemble the pathogen, and we ask their body to remember it. And that's how we've traditionally done vaccines. But as you know, in HIV, that has proven to be very, very difficult. And I'll summarize where we are with that. And so increasingly, the field is speaking more about passive immunization, which many people see as a sort of a plan B, but which has, as you will see, some potential. And in passive immunization, what we do is we bypass the immune system altogether, and we create antibodies, which are kind of guardians against pathogens. In this case, using antibodies against HIV, and we give them directly to people who are at risk, instead of asking them to make those antibodies through classic immunization. So let me tell you a little bit about where we are with both of these two concepts for HIV vaccine development. So the month has been a little sad in the sense that we have seen now published in the New England Journal of Medicine, finally, the data from what was called the Hptn 702 trial. And this was essentially a repeat to some extent of the RB144 trial, which did work, as you know, albeit at a low level, but it did work. Hptn 702 sought to perform essentially to test the same principle, but here in a different group of people in Africa. And perhaps with hindsight, maybe we changed too many things here. We changed the vaccine slightly. We changed the population in which we were testing the vaccines. We changed the adjuvant. We changed a lot of things. And unfortunately, for whichever one of these reasons, this vaccine failed to protect. And that's shown here. You can see that the red line, which is the vaccine, and the blue line, which is the sugar pill, are essentially overlapping. So no outcome here. And I think we'll spend a lot of time trying to understand in the next few months which part we changed that caused this vaccine to fail to work here. But as you heard from Cindra, there is an ongoing trial called Hptn 705 or Imbokodo or Mosaico, and that is testing a whole different concept altogether. This is in ways a very similar concept to some of the vaccines that have been tested for COVID. This is using a viral vector, sort of a Trojan horse, called adeno26. And as you heard from Cindra, it incorporates little elements of many different HIV viruses from across the globe. And the aim of that is to make it harder for the immune system to be able to get away from it, and easier for the immune system to see those bits of the virus that can't change. And this is at a very advanced stage, as you heard, and we'll have those data in the next year or two. So that's kind of where we are with in terms of active vaccination. But I'm going to spend a little more time telling you about some more upstream work. This is more developmental work rather than phase 3 clinical trials. So those of you who know me will know that all I can do is talk about broadly neutralizing antibodies. I spend a lot of time talking about them. So let me first start by explaining what I mean by broadly neutralizing antibodies. So anybody who is infected with HIV mounts a really good immune response to the virus. But the problem with most people's immune response is that the antibodies that they develop in response to infection are only able to recognize their own virus. And that's probably 80% of people. That's how their immune responses behave. But in about 1% of people, who we call elite neutralizers, develop the kinds of neutralizing antibodies that can recognize their own virus, but also can recognize viruses from across the world. And these are what we call broadly neutralizing antibodies. Neutralizing simply means they can stop the virus in its track, and broadly means that they can stop viruses from across the world. And of course, that's a game changer for prevention, because if we could either persuade the body's immune response to make these kinds of antibodies, or just give them to people to protect them in the short term, then that would be, as I say, a game changer. Over the last decade, many people have delved down into people's immune response in really huge detail and pulled out those individual B cells that are the ones that make the right antibodies, and separated them out from all the other antibodies producing cells in the body, and really understood at the molecular level what makes these antibodies special. And those are these, what I've shown here is these red antibodies over here, these Y-shaped molecules. And that's been really incredibly useful, because it's enabled us to, what I've said, define BNAB ontogeny as a template for vaccine design. And all that means is it means that we can try to copy what happens in people's own immune systems when they manage to make the right kinds of antibodies. And we can copy that in a vaccine strategy. And then the second thing is, as we've already mentioned quite a lot, is that we can use broadly neutralizing antibodies for passive immunization, both for prevention and, while we're not talking about it today, but also for treatment potentially. So we know a lot about, people like me have spent years studying this. We know a lot about how these broadly neutralizing antibodies, these ones in red here, develop. And essentially how they develop is through a long, and this is a problem, a long arms race that happens between the virus, shown here at the bottom, and the antibody at the top. And what happens is that the virus changes, and so the antibodies change to catch up with the virus, and then the virus change again, and the antibodies change again. And eventually, in some people, you end up with these broadly neutralizing antibodies. So that's taught us two important things as we think about designing vaccines. The first is that you do need some diversity. In infected people, it's that changing virus that drives the right antibodies. So it's possible, likely I would say, that if we want to drive these kinds of antibodies by vaccination, then our vaccines will also have to change a little bit. So instead of doing what we do with the flu vaccine, where we give, instead of doing what we do with the measles vaccine, where we give the same thing every time, we're going to have to do more what we do with the flu vaccine and come up with cleverer ways of doing it. But in this case, we're going to have to give them one after the other to try and replicate this process. The other thing we've learned is that these, what we call the germline BNAB precursor, which is just the ancestor of a broadly neutralizing antibody, they're very rare, and they often are quite funky. They have unusual features that set them apart from most other antibodies. And so you're looking for a needle in a haystack. If you want to get these guys going, you have to come up with clever ways to define the needle in the haystack and set it off along the right pathway. Checking for time. This has resulted in really three overlapping, and I'll explain how they're overlapping, three new concepts that have entered human clinical trials for the first time. And I think all of them are providing some interesting information. One of these is called germline targeting. And this specific approach is to go after those needles in the haystack. So these are heavily engineered proteins, as you can see from over here, that are specifically designed to find the needle in the haystack, pull it out, and get it going along the right pathway. So that's germline targeting. You will also have heard a lot about trimer immunizations. What trimer immunizations do is that they, again, use a very engineered protein that mimics the envelope, which is the bit that sits on the outside of the HIV particle. It's a notoriously difficult protein to work with. It falls apart and it's covered in sugars and it's just impossible to deal with. But people have worked very hard over the last decade to make it much more stable. And now it sits on top of these particles in a much more stable and well-replicated way. And that means that you can use it as a vaccine. So that's trimer immunizations. And then you will also have heard about sequential or lineage-based immunogens. And all that is what I've already mentioned to you, is that what you do here is you take a vaccine and you give one shot and then you change it ever so slightly and you give another shot and you change it again and you give another shot. And the hope here is that you can push those antibodies along the right pathway towards being broadly neutralizing antibodies. This is one of my favorite covers from a Cell paper, but much of this work that I'm going to talk about here, germline targeting, in just 2 slides for the organizers before they start having a heart attack, is where we are with germline targeting. And it's essentially, as I said, this is looking for the needle in the haystack, and then jumpstart it. This is what this wonderful cover shows here, is jumpstarting the right B cell and set it along the pathway. This is work that's really been led by Bill Sheaf, who's been very much in the news with this work, and refers to it as reverse vaccinology. And the whole premise of this is that all of the antibodies that happen to recognize this particular site called the CD4 binding site, which is really the focus of germline targeting, they all look virtually exactly the same. They all lock into the virus in almost exactly the same way. And it's that reproducibility that means that we can try to design these heavily engineered things that get them going. I think really exciting data that was shown at R4P is from a first phase 1 study of this thing, this immunogen called EOD-GT8, which has now been tested in 48 folks who received a high and a low dose, 2 doses. Very exciting, no safety concerns, but even more exciting is that 97% of these participants developed the right class of beginning cells. So when we talk about that needle in the haystack, they found that needle in the haystack and they set it off along the right pathway. And they did it commonly enough, this is what this last point speaks to, that there's a chance that we can find them again and set them off even further. So this is really exciting data. The other thing that I think is going to be very exciting, we are waiting for this data, is this use of trimers as immunogens. This is a slide that reminds you that these trimers have been tested exhaustively in many different animal models, rabbits and guinea pigs and monkeys. And they do generate antibodies. They may not be the perfect antibodies, but they're better than we've had for a very long time. And so this really provided the basis for doing this in humans. And that is exactly what has happened here, is that we've now started doing these in humans. And I think particularly as a scientist based in Africa, this is particularly exciting for me because it takes a trimer that was first identified in Africa in Kenya. And this study started, I think the slide shows, in 2001. And we've really come full circle and are now testing this vaccine that is based on this Kenyan virus in Africa. This is a trial called W001, or as my colleagues call it, WU1, and it's going to generate really exciting data in the next few months. Nobody is suggesting that the antibodies that it triggers will be broadly neutralizing. This is one step in the right direction, we hope. Not the magical solution to vaccines, but I think a step in the right direction. I just want to make the point that we tend to consider all of these things as separate entities. Germline targeting is separate to sequential, but they're all going to be the same in the end. We're going to probably need a vaccine that does germline targeting to get those needles in the haystack going. And that will probably be followed by sequential immunization, and probably the sequential immunization will use trimers. So we need to stop thinking of these as separate entities. They're all linked. Okay, so that's where I wanted to show you where we are with upstream studies to understand broadly neutralizing antibodies from an active vaccination perspective. But what about passive vaccination? I'm sure you've all heard of AMP. The results were released very recently. This took a broadly neutralizing antibody called VRC01 and put it into humans, into men and women who were at risk of becoming infected, and said, does it protect?
Hi, sorry, Penny, it's Sarah here. Just a note from the translators, if you could slow down the pace a bit, just because of the technical language, but I do recognize that you have just about a minute. In time. Cool. Yes, it is a challenge.
More slides, yeah, cool.
And a vote from me, you have 2 minutes.
I thought I was going to get that somewhere along the line. Okay, 2 minutes will do. Very slowly, though. So AMP tested the ability of this antibody to protect from infection, and we just got the results. And they are a little sobering, but they still do give us some hope. So essentially, what we saw here is that how well VRC01 worked depended on how sensitive the virus was that these people were exposed to. So sadly, if the virus wasn't very sensitive to the antibody we chose, you saw no effect, no protective efficacy at all. Even if the virus was only somewhat sensitive, you saw no protective efficacy. But if the virus that those people were exposed to were very sensitive to that monoclonal antibody, then in fact there was good protection, 75% protection. So what that means, it's been a complicated story to unravel, is that while overall this trial showed no protection, when you look at the most sensitive viruses, there was good protection. And what that means is that we can do passive immunization, but we need very, very potent, in other words, powerful, broadly neutralizing antibodies. One way we can do this, and this is my kind of last data slide, is by, instead of using just one antibody at a time, is by putting lots of antibodies together. And that increases the breadth and the potency quite substantially. So if you look at the gray lines here, this is what happens when we have one single broadly neutralizing antibody. But you can see that when we add two antibodies together, the red and the blue lines, we get much, essentially the line goes up, and that shows you that you have much better protection against viruses. And I think this is the way that the passive immunization field will move. And that is really the conclusion of my talk, which is that I think we are in a new era for broadly neutralizing antibodies. This is one of my favorite slides because I think it really highlights the role of the community. So if we didn't have these people who had developed these broadly neutralizing antibodies and they weren't prepared to allow us to study them, then we wouldn't be able to come up with these cool new concepts for active immunization. And we also wouldn't have been able to pull monoclonal antibodies, these special antibodies, out of these people. And we wouldn't be able to put that antibody back into people. And so it's really only because of these incredible people who give again and again and again that allow scientists like me to do our work that I think we are in a new era for broadly neutralizing antibodies. And I think it's exciting times looking forward. And there I will end. Sorry for going so fast.
That's fantastic, Penny. You're trying to explain some extremely complex concepts, and I think you explained them brilliantly. If people have questions about it, please do ask to put them into the chat. I've put one in myself, and we'll talk about them in the panel. I think I'd like to move on to Michael and Ferran from Barcelona Checkpoint, who will be tying together the issue of access and of equity in and making sure that people do benefit from these new technologies. I think we've seen that issue come up yet again in the issue of COVID, of course. And so it's something we have to revisit seemingly every time a new prevention or treatment modality comes along. So let's make sure that we do start advocating for equal access to these exciting new things. Anyway, I'll let Michael and Ferran say more about this.
Yes, thank you very much, Gus. I hope you can see my slides.
We can.
Okay, that's great. So Ferran Pujol and me, Michael Meullbroek, we are from Barcelona Checkpoint, and we opened in 2017 Barcelona Prep Point, and we're going to talk about advocacy and access to PrEP on a community level. Well, this is quite a broad issue, and it includes a lot of points. So I'm not sure how I move forward now. Oh, yes. So I have divided in 3 parts, which could be looked at separately, but I think they are also interrelated, and we want to focus on the discussion at the end of this webinar. So I just wanted to mention some of the points and concentrate on the access of on Prep Continuum of care, and then some points on long-acting agents. So I have to go back in history. So we have to go back to the evidence. We had evidence about that PrEP worked already in 2010. In 2012, it was approved by the FDA, but it wasn't until 2015 that there were 2 European studies, the PROUD and the HyperK, who were stopped because of the success of the results, that we saw some movement within Europe. And when that movement started probably in the AIDS Impact conference in July 2015, where there was a plenary where Bruno Speer of AIDS talked and mentioned, and this is the slide of his presentation, that if PrEP is not adopted in real life, then there will be publication of PrEP users, lay people, community guidelines issued. and that they would contact overseas pharmacies to buy a generic Truvada and give it to friends. So that was really an impact on that impact conference. And we saw that in November of that year, France was the first country outside of the US to approve PrEP. But we had a long way to go, and that passed by giving information and create awareness in our community. Campaigns, there were the Facebook sites, websites, community meetings, and one of the issues also was the language. In the US, people who used PrEP were stigmatized. So there's great campaigns from the Chicago AIDS Foundation who focused on keywords. We want to use sex-positive language, use pleasure, intimacy, love, lust, desire. And also Bob Grant, principal investigator of the IPREX study, who talked about that words like high-risk people, risk behavior, were in fact stigmatizing words. And we should use more inclusive words to stimulate people to look for those options to include in their prevention strategy. So what happens if you want to get an intervention approved? Then they always ask for cost effectiveness analysis. And those is a way to examine both the cost and the health outcomes of an intervention. And it estimates how much it costs to gain 1 unit of health outcome. Now, to do all those mathematics, you want to know the actual number of HIV infections or the HIV incidence. You want to know how much HIV infections you will be avoiding, the reduction of quality of life of people who are diagnosed with HIV, and then of course the cost of antiretroviral drugs for treatment and healthcare, and also of PrEP. Now, we already saw in 2015 that Valentina Camiano presented the cost effectiveness study. And you look at the red dot, if they talk about more than 6 sexual partners, it would be over the 80 coming years cost effective. But even if Truvada or PrEP would have a reduction of 50% of the cost, it would be more cost effective. And with an 80% reduction, it would even be cost saving. Our study presented in 2015 about the cohort of HIV negative MSM showed that although the HIV incidence was between 2 and 4%, that people who had more sexual relationships and STIs could have an HIV incidence of 24%. So with a very high chance of acquiring HIV infection. And if cost effectiveness studies are done, but they are not making a difference of approving the PrEP, then you always get to hear what will be the impact on the budget. So that's basically what's the price of the product and how many people would need that. And then you will do all the studies and show that it wouldn't be everybody on PrEP and the price could be coming down, but it doesn't always work that way either. So at the end, what we are facing most of the times is a kind of moralism or institutionalized homophobia. And moreover, in the last year, we had the COVID-19, which was in many cases used as an excuse for not approving PrEP, of not implementing PrEP, or not adequately financing programs if PrEP existed. Then the community moved on in their activisms, and through the UK, where it was permitted, they purchased PrEP online. And then for other European countries, this was shipped to their countries. And then a lot of times, like Dean Street, Prep Point, the community centers and doctors who were willing to support the community did the control and harm reduction of people buying PrEP on their own. So here is an image of the ECDC from October 2020, where you see the status of the implementation of PrEP in Europe. However, the first thing you notice, there is no access still in all of the countries of the EU or the EEA and the UK, which is now separated. And even if you see the dark blue, the dark green spots, for example, in Spain, there is no equity within the country. So after 1 year after approval of PrEP in Spain, people in the Balearic Islands or Canarian Islands still haven't access to PrEP. Only daily PrEP is approved. We don't have any other choices. And we still are using the high risk criteria, which includes a lot of people who may benefit of PrEP. There's also a lack of implementation studies, which are able to find the barriers that exist in the different countries. And what we need in that case is also culturally competent care. So ECDC did a needs assessment and estimated, and that's only MSM, that 500,000 men would want or need PrEP in the EU. And the longer the delay in the access, the more infections will occur. So if you imagine, we are talking about since 2012 or 2015, how many HIV infections could have been avoided. Now, how many people are using PrEP? And this is, I took these numbers from the Global PrEP Tracker from AVAC. And so according to these numbers, between 63 and 73,000 people in Europe are using PrEP. So that's around 12 or 14% only of people in need of PrEP. And that brings me also to another point, and that's the Prep Continuum, because it's not only getting access, PrEP is working if people are staying on PrEP. So we also want to know how many people are still using PrEP after 1 year. And this is a study presented at the International AIDS Conference last year from the Kaiser Permanente, and that you can see that of the 12,000 people, half only were still on PrEP 1 year later. Now, I did the same schedule as we have seen for Europe, and here you can see that 500,000 men would be in need in PrEP. We don't have any idea how many people of them are linked to care, how many are receiving a prescription. We know around 60, 70,000 people are have initiated PrEP, but we don't have any idea about how many people are still in PrEP after 1 year. So roughly we can see there's a cap of more than 90% in Europe. So the treatment cascade was a great tool for people living with HIV and for people with HIV, and also to put pressure on governments. So maybe we could think about the need of monitoring PrEP in this way for Europe, but also for countries, but not per country. Even within a country, it could be very different from one side to the other. And also it could be a great initiative for the fast city tracks. Just a couple of notes for long-acting agents. We have heard a lot about it in the previous two talks. So probably the next year, Kabbategravir will be approved, but then we will also facing some new challenges. So the first would be, of course, the price. Cindra already mentioned it. There was a study presented at the last CROI, at the 21st CROI, and that showed that if the price of generic, and that's in the US, if the price of generic Truvada would be the same, then it would be cost effective if Kabbategravir would be cost maximum $11,600, so only $3,000 more. But what we see, generic PrEP in Europe costs around 300 euros per year. So we are talking about really very different prices. And would VIV be ready to offer Kabbategravir at the lower prices? Now, we had this experience also with Gilead. If they would have lowered their price, probably the approbation and access to PrEP in Europe would have been faster. And will we still be using cost effectiveness analysis? Or would we use other methods like the multi-criteria decision analysis, where different stockholders in focus groups talking about situations are producing a report.
1 minute, Michael, okay?
Yeah, that's fine. So these methods are already used for treatment of long-acting Kabbategravir and rilpivirine for people living with HIV. So maybe it's something of the future. Then the second thing, long-term adverse effects, also a thing we have to take in mind. We know about the injection site reactions, but what about weight gain? We have we talked a lot about it for people living with HIV, but what about for PrEP use? Here you can see that in the 083 study, there was a significant greater weight increase than in the Truvada arm. And then also the testing policy. Cindra already mentioned it. Not all HIV tests provide accurate results. At the CROI, there was a study presented from Thailand where they showed that the third generation antibody test was more accurate than the fourth generation testing. And then what we in fact need is viral load testing, but that will be only feasible if it's easy and fast to use, if they can detect below 50 copies, and if it's cheap, of course. And then we have to think also about counseling of positive HIV results, so results of people who have become HIV much time before. And then the last 2 points I want to mention, let's think also about the risk of acquisition of HIV for people who want to think about on-off PrEP, because it's not the same as people living with HIV. And finally, we have seen with Kabbategravir, and we will see it also with the new long-actings, Europe is not a place to perform clinical trials, and that's also really a pain in the ass. I think we should have clinical trials in Europe, and we should also have implementation trials where people and the community can get involved in how to take that forward. And with this, I will end, if I'm on time, and give the word to Will and Gus. Thank you very much for your attention.
Okay, thank you. That's fantastic. That throws up, I think, every sort of issue around access, equity, and the implications of long-term PrEP, including that really interesting last one about PrEP, about how it may change HIV testing and how we need to do it. I'm now going to hand over to Will of PrEPster, who is basically going to facilitate the panel discussion. And we have half an hour. I'll leave it up to you how to divvy the time up between the panelists and the people asking questions. So over to you, Will.
Thank you.
Don't forget to introduce yourself.
Thank you very much, Gus. I'm Will Nutland. I'm the co-founder of PrEPster. I am based in London, very much still European and part of Europe. As I always say, my huge apologies for all of the absolute fuckery that people in my country have done to do so much damage to the great relationship that many of us feel like we had with our friends on the European continent and beyond. With the politics of Europe aside, if you do have any questions that you want to ask any of the panelists, and actually maybe if the three panelists could turn their cameras on, that would be amazing, and your mic, so that we can dive straight in. So if you want to ask questions as they come up, we'll attempt to get through as many of them as possible. But just to get the ball rolling, I actually have a question for Cindra. So Cindra, there are going to be a lot of educators and a lot of advocates in today's webinar. And I know one of the questions that I get from commissioners and from policymakers when I try to persuade them to think about investing in these new pipeline technologies is, well, we've got oral PrEP. It works really well. Why should we invest and focus on these new pipeline technologies? And I know you've kind of addressed this in your presentation, but maybe kind of top headline hints for the advocates in the room about the arguments that we might use.
Well, I think I showed that, okay, I mean, oral PrEP is amazing. It's almost 100% effective. It works well, but it only works well if you take it as prescribed, if you take it every day or at the time of risk. And that's a heavy burden for a lot of people who cannot or don't want to take a pill every day. And so a longer acting device like a vaginal ring that you just insert once a month will get you away from these adherence burdens. They're also more discreet. A vaginal ring, no one has to know about it. The sexual partner doesn't have to know it, as with oral PrEP, but you're not carrying it around. The pills are not shaking in your bag. You don't need to take it every day where someone's going to maybe see you take it. And you know what? Maybe someone does want to take pills for 1 month, and then, or maybe someone wants to take pills for a week because they're going on vacation, or they're having a fling for a week, and then they stop, and then they want to go back to the vaginal ring. No one product is going to be the perfect product for someone their whole life. So the more we have, the more covered sex acts there will be.
Great. Michael, I'm also going to ask you a quick question, if I may. I know a couple of questions are already coming up in the chat box, and we'll move on to them next. So, Michael, thinking about the service that's offered right now from Barcelona Checkpoint, to what extent do you think services like yours will be geared up to provide these new technologies if and when they become approved? So if I was living in Barcelona right now, I could come and get my daily oral PrEP from you. Do you envisage that your service in 3 or 4 years' time, I could rock up and replace my daily oral PrEP with an injection?
Yes, let me just start by saying that when we started the Prep Point, it was a way to move forward. That was our first step. I mean, if we couldn't show that PrEP was already delivered to the community, then it would be more difficult to get PrEP approved in Spain. So this was one issue we had in mind. The second issue was, once it was approved, you saw all these hospitals not doing anything. They didn't do anything. So what we were doing, we're pushing the uptake of PrEP. And then slowly you see the hospitals around us, in comparison to other parts of Spain, are increasing the number of people on PrEP. Whereas in other parts of Spain, they still have to think about how to do that. So we are pushing by doing it ourselves, we're pushing the other ones. And so to get back to your question, well, injectables would be exactly the same. We are open from 8 in the morning till 8 in the evening. If you go to a hospital, you have to be there between 8 and 2. To get an injection, we already do vaccination. So to get an infection, we have already the nurses there to do it. So we have the fridge, we have everything there to do, and we have the opening hours. We have a culturally competent staff. So my answer would be yes, we can do it.
Okay, thank you. So I'm going to, we've got a question here from Ukraine, and let's see who might be the best person to respond to this. So this is about the vaginal rings. My foundation is working in PrEP for women now, and I think the vaginal rings would be one of the most effective strategies for Ukrainian women. So my questions are, when will these rings be available in Ukraine, if anyone of us knows the answer? What about the most common side effects? I've read a paper that in some cases there could be a risk of cervical cancer. And then thirdly, do we know how much the rings will cost per year, and will be generic rings available? Cindra, Penny, Penny, no? I'll come. Okay, I have a question for you in a second, then Penny, that's coming up. Cindra, is this anything that you can help?
I mean, I could take a shot at some of it. So like I had said, the EMA, the European Medicines Agency, did not approve it for Europe. For IPM, is the International Partners for Microbicides, they're the manufacturers, they own the patent. You would have to convince them to spend the money to go to Ukrainian regulatory to actually pass, to regulate it and say, yes, we think our country would benefit for it, but that's going to cost them, IPM, a lot of money to do that. But it's worth a shot to go straight to the manufacturers and ask them, say that there's, mobilize the community and say there's interest here. The common side effects in the studies, obviously it's Depiving Ring. I don't know that there's any known cancer side effects for Depiving, which is the ARV. Perhaps what you read was because the vaginal ring exists for pregnancy prevention, for oral contraception. So maybe that was the affiliation with the cancer. The side effects are very minimal. However, they can cause urinary tract infections. And if you've ever had a UTI, that's not a minimal side effect. That is a fearsome, horrible side effect. So I'm saying that wherever these are rolled out, they should be rolled out with UTI treatment or prevention.
And actually, Cindra, I just want to jump in here, because I think some of the conversations we've certainly been having with activists in London is just in the same way that when these technologies became unaffordable, we found generic versions of them in different parts of the world, or if they weren't improved in our countries or continents, we were getting drugs and importing generics around. It sounds like even if the ring became available in a cheap format, there actually might be some quite potentially dangerous side effects of people self-use, self-medicating with it, given what you've just said about uterine tract infections, for example.
Yeah, I mean, the great thing about it is that it is user-friendly. So once you're trained on how to insert it, it almost delinks you from the medical system to be able to, maybe pick it up at your pharmacy and not have to go to a doctor every month. I love the idea of a generic ring. I mean, because again, they already make it for, they make them for contraception. So they're probably available generically.
Okay, let's hold off on rings, see if we've got a bit more time to come back to them in a second. But Penny, a question from your presentation that Gus posed. Gus asked, there was excitement in the press about the AV vaccine trial in monkeys recently using an RNA vaccine. Can you say, Penny, a little more about that, and also in general about whether the drive for a COVID vaccine will help HIV vaccine development? Boy, that's probably a question that you need an hour to answer, but let's see if we can do it in 2 minutes.
Short line, I sincerely hope so. It should. Let me go back to the RNA question first. This was a cool study. It was presented at CROI. But essentially, it's using the same kind of technology as Pfizer and Moderna are using for COVID vaccines. And so I told you all about SOSIP trimers. Essentially, what these guys did was to take an RNA that would produce that SOSIP trimer and use the same technology as Pfizer and Moderna and to put it into monkeys. And they're essentially, instead of putting a protein in, you're giving them the RNA and asking the monkey to produce the trimer, which it did very well, and then asking the immune system to see it and to make antibodies. And in this particular case, the monkeys did make antibodies. In fact, they made pretty good antibodies, some of them with breadth, which is very exciting. And those antibodies were able to protect a subset of them from infection for a period of time. So it's one of the, I guess it's one of the first, to get back to the COVID question, it's one of the first direct links we've seen, where we're taking technologies that were warp speeded, to use the American term, and put into, and to go back to HIV and to translate some of those technologies backwards. But it's a two-way street, I guess, because I think a lot of the COVID response, a lot of the success of the COVID vaccine response is built on years and years and years of HIV vaccine response. So we're now seeing that translated into a pandemic response. And what I really hope is that the realization that you can translate that into a pandemic response means that we'll rethink HIV as a pandemic again, instead of, this other problem that we have. So yeah, short answer, I really hope so, it should.
Amazing. And so some quite exciting potential developments there, things to get excited about. So we do have another question about Kabbategravir injection that I'm going to hold off on just a second, because as I mentioned at the beginning of this section, are a lot of educators and advocates attending today's session. And I really want to make sure that we're starting these conversations about how educators and advocates across Europe can be equipped to start advocating for these pipeline drugs to move, from test tubes, from the labs, from phase 1, 2, and 3 clinical trials actually into people's bodies. And so, loads of people attending today's webinar have been fundamentally involved in the push for oral PrEP, whether it's in Europe or outside of Europe. And so I want to ask all three of the panelists, what can we learn from the advocacy that pushed for daily oral PrEP that we can translate over into the advocacy for all of these pipeline PrEP author and vaccines as well. And whoever wants to go first, I'm happy to let you start. But I want all three of you just to touch on that question. What are the advocacy things that we can learn from? And Michael, you've got your hand up, so let's go with you first. Hey.
Yeah, very quickly, I think one of the important issues what we saw is that just moving forward was an important step. Just to have PrEP available, buying it, people getting information, just doing it, being a step ahead of the healthcare providers even and the government, and so that's one thing. And the other is, within that context, I think it's also important that we had a possibility to start within a clinical trial. This was also to have a clinical trial here. For example, if Kabbategravir would have some sites in Europe, you could already show that it's working, that's in place, that they're already doing, and that's helpful for people to make the other step. So please go ahead.
Absolutely. Which was exactly the model that was used in England with the PROUD trial and then the IMPACT trial. So Cindra, that's I'm sure you've got a whole load of hints for us advocates.
Just really, really concisely, and learning from PrEP. Coordinated pilot rollout, but less pilots. As Linda Gale Becker, a great researcher, said, we all have pilotitis. So not every country needs to roll it out, implement a pilot program for a year or two to see how to implement it. You know, we can have coordinated pilots across the globe. Okay, let's do it in gay men in these 10 countries. Let's do it in young women in these countries, and boom, let's get our information immediately. That's one. Another one that it really took off in the US when gay men just took to social media and really it spread like wildfire. And generics, keep the prices down, generic competition.
Penny, anything to add to this?
Yeah, so I guess I'm not an advocate. I'm a scientist who occasionally advocates. You know, for me, I think what we've learned is that scientifically there's a good reason for us to be testing vaccines in different populations. You know, that data is clear. So we need to involve everybody in the clinical trial testing effort. And then I think the other thing that we need to be less scared about, and we are scared as scientists, we're very scared of this. We're scared of experimental medicine. And I suspect it's not just the scientists who are scared of that term, experimental medicine, because it's a terrible term. It kind of makes humans sound like guinea pigs. But actually what it is doing, and maybe it's like pilots for vaccines, what it is doing small scale trials safely and quickly, rather than waiting for the large scale things. You know, we are learning such a huge amount through these little tiny trials in people. That is much more applicable to real life and you know, scientifically valid and providing these huge insights. And maybe we need to come up with a much better phrase than experimental medicine, which terrifies even me as a scientist. It's a terrible phrase, but we need to be doing more of that kind of science. And I think we need to be pushing our scientists, people like me, who have ideas, but don't see them as being as we don't get to the next step fast enough. People like me don't go fast enough. You know, we're too hooked up on the scientific ideas, and I think we need to get them into people faster, safely, obviously safely. But we need to get those answers from people and from different populations, and we need to move faster as scientists.
Fantastic response. I would argue that you are an advocate and that all scientists are advocates, but that's a discussion for another day. I've got a couple of quick things I'm just going to throw into the mix here because there's some questions from
Ferran, did you want to add something? Oh, sorry.
No. Ferran? No? No, okay. So I want to just run back up. We have a Kabbategravir injection question from Kim. Cindra, I think you might have touched on this in your presentation. Kim's asking, I just want to know if the Kabbategravir injection has no side effects with silicone injections or implants. Is this something you can help us with, Cindra? You're on mute.
Okay, sorry.
Okay.
So unfortunately, the inclusion criteria for the studies, they allowed trans women in the study. However, they could not participate if they had buttocks implants. So we don't know if there is a contraindication if something bad happens with buttocks implants. However, if you look at all of the other PrEP formulations like TAF and TDF-FTC, and there was a number of trans people. There was about 12% trans representation in this study, and they didn't find any hormonal ARV contraindication. So it was all it was fine as far. It didn't deplete the amount of hormones, and it didn't necessarily deplete the clinically matter. It didn't show any difference between trans people and MSM or gay people. The trends were the same. Now they didn't have enough trans people to do a statistically relevant or significant comparison, but their trends they saw were the same in MSM as trans women. So it's safe and effective.
Okay. So we are just coming down to about 3 or 4 minutes left. I know I'm going to take one question from Roger at NAM, and then if we have one final question, and a quick one from someone who hasn't yet asked a question, then if we've got time, we'll take that. So if Roger from NAM says, clearly nothing official has been said about the pricing of Kabbategravir for PrEP. What does the panel suspect or know anecdotally about ViV's intentions in respect of price? Does anyone have any sense of this? Cindra?
I can only guess. I mean, it's pharma, so just look at their history, be careful, be out in front of it, push for generics.
Yeah. Michael or Penny, anything more to add?
Well, I asked them directly, and so, well, the Spanish part of the company, and they said, well, they are still preparing their dossiers, and they really can't say anything about it. That's still the official standpoint.
I wrote up, I put things, references in the Q&A to various cost-effectiveness studies and things like that, but I would like to comment on just on the one. There's an American cost-effectiveness study which did look at the price of Kabbategravir rilpivirine as treatment. It took the sort of worst case scenario, it took the list price of Kabbategravir rilpivirine, which is very high in the US, and basically said how much would we have to scale this down in order for it to be cost effective. And there was a sort of sliding scale, and that implied that because of the extra bonus you get from good adherence, it could be more expensive than oral PrEP, but not a lot more. It would probably become cost saving if it was within the region of 2,000 a year more expensive than oral PrEP. But that would be in the high income world. In the low income world, it will really have to be very cheap to be cost effective.
Thank you, Gus, for adding to that part of the conversation for us. So we are right on time to actually move back to Gus in just a couple of seconds. Thank you, Penny, Michael, Cindra, for fielding those questions. Thank you for those people who fired questions into the chat box. You made it very easy for me to manage that section. I don't think we have any unanswered questions within that, and thank you for the folks who are translating those questions over to Russian for us. Gus, thank you, back over to you.
Yes, thanks very much to everybody, including you, Will, for facilitating it so well, and particularly to Sarah, who really put a lot of the energy into this, and who I will hand over to for the last minute bits on things like publication, stuff like that, and on the evaluation form. I would just thank all the presenters. Look at the chat box, because people like me and also Mitch from AVAC and so on, I see, were sort of feeding in additional information, because there's always too much to say on these things. Thank you very much, and I hope this really stimulates more discussion, and especially on issues of access and pricing, which I think as a community we need to continue ramping up on. So, Sarah, I'm going to hand it over to you for the last word.
Hi. I wasn't expecting that, but but that's okay. No, it's pretty easy, because everything has really been said. So yeah, big thank you to everyone who attended and joined us today. And yes, so we will be sending out the PowerPoint slides to all of you who attended. They will only be in English, but we will be sending out a feedback form to attendees, which will be bilingual. Because we would love to hear how we can make this even more accessible, and to all NGOs and community level advocates and educators across Europe. And oh, thank you.
And the recording?
I think that was it. Oh yes, the recording. So as we mentioned in the event reminder and at the beginning of this meeting, it is recorded, both the English and the Russian channels. So at a later date, you can expect to see this recording either on the EATG, PrEPster, AVAC, Prep in Europe social media platforms. But that would be further down the line. But if there were any concerns around confidentiality, you can get in touch with me. I believe everyone has my email. And I will be sending out the feedback forms and PowerPoints. But I think that's everything. Am I forgetting anything? Organizing committee, thank you to Gus and Cindra, Anis, Will, for helping make today happen. And Penny, and Michael, and Ferran, of course. So many.
Yep. Okay. Thank you for everybody. I'm going to put in a plug for the next Prep in Europe one on May the 15th about PrEP and women. There are other EATG ones coming along. It's all very exciting at the moment. So I'm going to sign off. But Sarah, you have to press the magic leave button because you're the host that ends it.